welcome. Welcome to this first talk in a new series that we've started this year in the spirit of making sure we communicate better across and within the Institute to make sure people have the opportunity to learn about a lot of the exciting research going on here. You know, we spend so much time inviting people in from the outside and hearing about their work. We really don't spend enough time hearing about the great work going on here at the Institute. So as part of a series of conversations this year, the faculty decided it would be a great opportunity to add another series of talks. We're trying to do this at a time. Normally we'd be having one type of a faculty meeting or another, but Wednesday at lunchtime that hopefully won't be too disruptive to people, but give everybody the opportunity to learn more about what our colleagues are doing. So I appreciate everybody being on, and I'm very appreciative of Carla Finkielstein, who will be our inaugural speaker in this series that we call the Primary Faculty Research Program Update Series. And so I think most everybody knows Carla, but I think a lot of you probably here know Carla by virtue of all the exciting things you've been hearing about in terms of the COVID testing at the Molecular Diagnostics Lab. And believe it or not, even though Carla spends about 25 hours a day working on that, she actually has another life, although I don't know, her family may argue with that at this point, but she has this other life in science that relates to her work on cancer, and particularly on breast cancer, and some really exciting things, understanding molecular clocks and how they interface with the possibilities for therapeutics and ways of potentially treating cancer. So I think, as most of you know, Carla is one of the newest members to join the FPRI. She is an associate professor in biological sciences in the College of Science and here at the Research Institute. And today she's going to tell us about her work on exploiting molecular crosstalk mechanisms for cancer treatment. So Carla, welcome. Thanks for doing this, and I'll turn it over to you.
Thank you very much, Mike. Thanks a lot for the introduction. Let me share my screen with all of you. Okay, Brad, is this okay? Everybody can see this?
Yeah, you're good.
Okay, excellent. Well, thank you very much for joining us and for sharing your lunch with me today. I had to confess that I'm much better when I walk around during my talk, so talking to my computer to a virtual audience is not what I call the high of my day. But since we are in the COVID time, it is what it is. So let's start with the most relevant slide, which is the disclosure. So as you all know, we all need to start our talks with a disclosure, and my disclosure is very simple. This talk will not give any credit to any punk with 5 genes that has recently been in the news. The upper right corner that you actually see here has the unofficial logo of our lab in Riverside 4. And I hope all the girls that are listening to this talk that they're in Riverside 4 already finished the neutralization of all the samples today. I also want to mention that the word that starts with C and finish on 19 will not be mentioned in my talk today. So if you expect any of these 2 words mentioned today in any way, I will disappoint you. And the last thing that I want to add in my disclosure is that, let's be honest about something. If this pandemic would have been called the stay-at-home challenge and posted in Facebook, the virus could be gone by now. We all know that this is true. Now, being really honest about absolutely anything, I hope that you actually had the chance to read the latest article published in Nature Medicine last week. And if you haven't done so, please do it. It's really worth your time. In this article, they actually model all potential scenarios for which this disease can spread over the United States. And one of the most interesting conclusions of this is that if 90% of the population of the United States wears masks, we can save 130,000 lives from now to February next year. So the question that we all ask is, why is that masks are not mandatory? Okay, maybe we should follow the lead of places like Vietnam or Uruguay, where mask is mandatory and the rate of death is very low. Okay, done with this guy. Let's talk about science and what we do in my lab, why we do it, and where we're going. So the most important thing that I want to share with you today is that what we are truly passionate about in my lab is this connection between circadian rhythms and the cell cycle. And one of the reasons that drove my attention to this connection is the fact that mammalian cells, they have this very interesting transitions, or this very interesting cycle where the cells divide, and then go to a resting stage and duplicate the DNA and follow the cycle in every 24 hours. And that each phase of this particular cycle follows a very specific timing. For example, cells enter a resting phase and a phase of accumulation of nutrients during the night from around 11 p.m. to 8 in the morning. From 8 in the morning to around 5 p.m., the cells duplicate their DNA and everything else that is in the cell, including organelles. And then towards the end of the day, around 5 or 6 p.m., the cells enter in what is called a G2 phase or GAP2 phase of the cell cycle, where they actually control that everything has been faithfully duplicated. And if so, at 10 p.m. sharp, they enter a phase of mitosis that lasts for 1 hour. And after that, the daughter cells split and enter in a resting phase again. What is particularly interesting about this is the fact that it's so timely regulated. So that talks about a connection between the cell cycle and the mechanisms that control the time inside the cell. And that is what is called the circadian clock, which actually brings me back to one of my favorite physicists, Christian Huygens, who centuries ago actually brought to our attention the fact that two clocks, that they are mechanically connected, tick-tock eventually in synchrony. And this is what you actually saw probably in the little movie that I had down here. So why is this important? Because when cells, they don't synchronize their external cues to the division cycle, is when very many of the disease process emerges. So there are a number of questions that we try to answer in my lab. Some of them are basic questions. Some of them I expect eventually will be more on the translational arena. So the big question that we try to answer in my lab is, how do cells sense time cues? How is that the cell know that 10 p.m. they need to enter mitosis, okay? How do cells adjust, reset, and reprogram their metabolic and genetic makeup to actually changes in those environmental cues? And more importantly, every single cell in the tissue has a mini clock. So this cell here and this cell here, they need to connect, and they need to talk, and they need to do it at exactly the same time. So how is those intercellular coupling occurs? Now, when we move to a little bit more, when we depart a little bit from the basic questions and we move towards a little bit more of the translational arena, we try to answer other important questions, which are, or at least important to us. So could it be possible that disruption of this circadian system, the circadian cell cycle system, be responsible for some of the proliferative disorders that we observe, like cancer? And then, is there such a thing that a different clock in a cancer cell than in a normal cell? Is that possible? And more importantly, and this is what I want to convey to you, is there any possibility that we can use a time of day approach to either determine which one is the best time to deliver a drug or to treat a patient? So all these kind of things, really, all these kind of questions arise from years of work by my colleagues in the field of circadian rhythms. And that really started in early 20s and received a lot of attention early on the 70s and 80s, and more recently by the work of some of the really hardcore molecular biologists that put basis for the understanding of how this rhythm operate. So early on, a number of studies established that the suprachiasmatic nuclei in the brain is the structure where the master clock is located. And the master clock senses light or some environmental cues and generates an output signal, sometimes in the form of the hormone, that actually talk to peripheral organs and synchronize what is called slave oscillators. Those slave oscillators have one particular job, that is synchronize the whole body to the external cue. Now, these structures were determined early on, and of course there was a rush to determine the molecular basis for this, for the way that these clocks operate. And that brought about the work of Kanopke, that early on, he determined using forward genetics that there's a gene that is involved in controlling the rhythmicity, the pace of the clock. And this gene was later named period 2. And a great part of the studies on the circadian clock at the molecular level was done by 3 adventurous scientists that received the Nobel Prize in Physiology and Medicine in 2017. 2 of them, Jeffrey Hall, here on the left, and Mike Rorschach, discovered that period 2 was a negative regulator of the clock. And their work basically opened the door to the discovery of the transcription and translational feedback loop that controls the progression of the clock mechanism. Now, the same way that I'm going to use the same sentence that the Nobel Committee used when they described the work of Michael Young, the devil is on the details. Michael Young was the one scientist that actually discovered that transcription and translational feedback loop was controlled mainly by post-translational modifications, particularly phosphorylation by casein kinase 1 epsilon. I just want to bring to your attention that Michael Young will be here at FBRI in April next year, and hope all of you and myself are vaccinated by then. And he will be giving a talk exactly on this particular topic. So I encourage you to go. He's a fantastic speaker. So anyway, why is all this important? Because nowadays we know how the molecular clock works. So at the beginning, we have a lot of information on the physiology of the clock, but we didn't have much of molecular understanding of how the clock works. And the last 15 years has been beautiful in the research of clock mechanisms, because we found out all these molecules that play these roles in controlling different processes in the cells. We know that the core clock component is a dimer called clock and BMAL. And this clock BMAL dimer controls the expression of negative regulators like the period and cry proteins, which actually, depending on where they express, for how long they stay, and what post-translational modifications they have, feedback back to the core mechanism and negative control the expression of clock BMAL. Clock BMAL also control the transcription of other genes that are called clock control genes. These clock control genes, they are not part of the core clock mechanism, but they regulate other processes involved in multiple things. Just pick any of your favorite processes, and I challenge you, you will find at least one gene that is circadian regulated. So the important thing about these clock mechanisms is that they really control a great number of physiological functions, okay? Everything from your sleep-wake pattern, your body temperature, blood pressure, just name it. Basically, everything that it goes at the physiological level is somehow controlled by the clock mechanism. This is particularly interesting because, as you can imagine, the power that this clock system has is tremendously, and any deregulation in any of the genes that control this machinery could render in the disease. And in fact, we see nowadays that a number of diseases and disorders emerge when the connection between the clock system, the master clock, and the slave oscillators in organs is not working well, okay? So this could happen because they don't talk to each other well, or they could happen because one of them is completely deregulated. And I did a quick search the other day in Google Scholar, and I keep finding more and more diseases associated with this function of the clock mechanisms. And some of them are kind of, intuitive and obvious, like obesity, for example. There's a lot of work done in metabolism and circadian clock. And if someone is interested, I can talk about the circadian diet, which is a really cool diet to follow, especially because it allows you to eat basically everything that you want. The only constraint is the time of the day at which you have to do it. But from the standpoint of my lab, the important disease that has been somehow linked to clock dysfunction is cancer. And one of the earliest discoveries that cancer is linked to the clock, well, I apologize for this little thing here. I just noticed there is a fly on my white screen. It has to do with this fantastic epidemiological prospective study that was done, that started actually in 1976 and continues today, and enrolled 275,000 nurses, female and male nurses, that has been followed over this almost 50 years by now, and that shed light on the connection between night shift work and carcinogenicity. So we know now that women's working night shift, they have 37% higher propensity to develop breast cancer. More recently, a study shows that male, they also develop prostate cancer, and some new evidence shows that there are colorectal cancer associated to night shift work. This is particularly important because night shift is really the main source of work hours in healthcare, manufacturing, retail service, and transport. I mean, flight attendants, they do experience already a lot of displaced rhythmicity because, or misalignment because of their time zone frequency fly. But on top of that, the fact that they're working on unscheduled hours makes their work more dangerous. So the International Agency for Research on Cancer has labeled night shift as potential carcinogenic, okay, or probably a carcinogenic. And this is based on 3 parameters. One, limited evidence of cancer in humans. Despite 275,000 women having followed every 50 years, still, we need more research on this. So this is kind of a prevention. Sufficient evidence of cancer in experimental models, and strong mechanistic evidences. So because of this, very many European countries have banned chronic night shift work from their workers. I mean, there are policies in France, Sweden, and UK that prevent workers to work more than five years night shift. And there are other policies that prevent workers to alternate night shifts during the week, because that completely misalign the clock. So as you can imagine, the night shift work is certainly an issue, especially if you consider that 80% of the world population is exposed to light at night, and 20% of Europeans and 29% of American workers work actually night shifts. Okay, so the easy solution for this is really to prevent long-term night shift work, okay? So that's the easiest way to do it, and Europeans have been very proactive on this aspect. So one thing that I wanna mention is that I was really puzzled by these findings, and we run a number of studies with my colleagues from the University of Buenos Aires in Argentina, and the reason because we team up is because they are very well-renowned circadian scientists that study the immune system. And I challenged them to test whether there is a possibility that the misalignment of the circadian clock in animals that has been challenged with cancer cells could result in tumor growth due to a compromised immune system. So to do that experiment, we actually synchronize animals to 2 different schedules. One is a light-dark schedule where 12 hours are light and 12 hours are dark. And then another set of animals were synchronized for 3 weeks in what is called a chronic jet lag schedule, in which basically we advance the circadian time for 6 hours every 2 days. And this creates a chronic jet lag that is equivalent to flying 21 flight zones. Okay, after 3 weeks of maintaining the animals in this schedule or in the normal light-dark schedule, we challenge the animals with melanoma cells from the B6F10 type, which is a non-metastatic melanoma cells. And we chose these ones in particular because we know that tumor growth of those cells depends on the immune environment. And we follow the animals We maintain the animals on their initial rhythm for a couple of weeks, and we follow them over time. One thing that we found that was particularly relevant, and I don't want to go over all the results because it's not the purpose of this talk and they're published already, but one thing that I want to bring to your attention is we observe immediately that we run this experiment two phenomena. One, a very short latency, okay? So latency divided as the moment in which, from the moment that we inject the cancer cells to the moment that we detect the first tumor, we can pulp the tumor, okay? So there's a very short latency for those in those animals that actually were circadian disrupted. And the other thing that we notice is that by the time that we sacrifice the animals, okay, the volume of the tumor in the animals that they were disrupted, they have a circadian disruption, was 4 or 5 times bigger. It was a dramatic difference. This got our attention, and we certainly ran a number of additional experiments that went in 2 dimensions. One was all about the tumor and study how the cell cycle in the tumor was somehow influenced by the circadian disruption. And the other part was all about the tumor microenvironment and how the immune microenvironment actually was totally remodeled to favor tumor growth. And we actually found that, for example, macrophages, they were expressed in completely anti-phase that we were normally expected in these animals versus the one that they were circadian desynchronized. For example, in these animals, macrophages of the type M1 are usually present or increased during the night and reduced during the day, and M2 macrophages, which is another type of immune cell, follows an anti-phase expression. What we found is that in animals that they have this tumor deregulated, either the macrophages were not oscillated anymore, or they were at very low levels. We also found differences in the expression of interleukin cells. We found differences in the numbers of LT-REG cells, and all of them brought us to the conclusion that there is a huge dysregulation in the immune system that is not only in the tumor microenvironment, but was actually shown later on to be also compromised in peripheral tissues. We also find out that you can see here that the clock itself was deregulated in the peripheral tissues of some of these animals, okay? And you see this because we monitor the expression of the BMAL and the CRY proteins, transcripts, and you can see that in animals that were subjected to chronic jet lag, the peripheral organs, they actually lost completely rhythmicity. We also find out that a number of cell cycle genes, and I'm just going to point to only two of them for simplicity, are completely deregulated in animals that they are circadian compromised. And this happened in 2 different tissues, in the tumor itself and in the peripheral tissues, suggesting that somehow there is a complete remodeling of the cell cycle and the immune system right around where the tumor was injected, and that favors tumor growth. This is particularly remarkable if you really think that immunosuppression, chronic inflammation, and cell proliferation are all phenomena that are clearly regulated by the clock. So, but which ones are the molecular basis by which all these phenomena happen and take place? Well, we know nowadays that animals that had been knocked down for the expression of circadian genes, or in other cases where circadian genes were overexpressed, they do show a phenotype in which there is a clear change on the cell cycle, or that causes like abnormal growth or early aging. So we found, for example, that, let me go back. it wasn't our work, but our colleagues found that when you knock down PER2, for example, the animal, of course, goes to sleep early or wake up late. I mean, it has some kind of deregulation of the circadian rhythm. But on top of that, these animals develop spontaneous cancers. So there is a clear connection at the molecular level between several of these molecules that control the clock mechanism and those that control the transitions on the cell cycle. So the question that we ask in our lab is, how do we find out which ones are those connections, how they are connected, and which ones are those molecules that talk to each other and control these transitions. And in my lab, we are expert in analyzing cell signaling events, and we are experts in connecting the dots between one molecule and the other one. That's what we do. That is really our strength. We are really signaling people. So for us, the best example of how signaling works in a cell is probably the work of Jackson Pollock. This is a famous N14 gray canvas from Jackson Pollock. And I think it really represents signaling events very well. I mean, this is what we normally see as signaling events in the cell. You can actually, if you put a little bit of thought in this canvas, you can actually see nodes of regulation. And you can see a lot of feedback loops here. So this is the way that we actually see the problem inside the cell. And that's why you're probably gonna talk to molecular biologists like me. And when you bring about problems that involve two cells, we freak out because one cell for us is complicated enough. Two cells is a world. And an animal is absolutely crazy for us. So anyway, so what we do in the lab basically is untangle this mesh. That's what we do. We just try to untangle absolutely everything that is here and find the real connections. And I'm not gonna go through all the experiments that we have done to actually untangle things. I'm just gonna tell you the end of the story because you can see our publications. But basically, a couple of things that we discovered is that molecule PER2 or period 2 that controls circadian rhythms happen to associate with a molecule called P53, which is a tumor suppressor that is mutated in most of the cancers. Not only that, when PER2 associates to P53, this association prevents P53 degradation and ubiquitination by the negative regulator of P53, which is the oncogenic protein MDM2. Later on, we find out that PER2 interacts with P53 and blocks some signaling events. And this blockage of signaling events help to control the response to genotoxic stress and DNA damage in the cell. Something that is very important if you consider that most chemotherapeutic drugs and radiations is what is used for the treatment of cancer. So the second piece of evidence is that we added to the model is the finding that PER2 and P53, when associated, control the transcriptional activity of the tumor suppressor. So if PER2 is bound to P53, stabilizes P53, but P53 cannot be transcriptionally active. So in order for this tumor suppressor to do the job, then PER2 cannot be associated to. So when we were very happy about this, we run into an experiment that was troublesome at the beginning, in which we found that PER2 and P53 didn't coincide in in time. And that was kind of troublesome because we published several papers clearly showing that PER2 and P53 stabilize each other. And of course, if that is the case, you would expect that when this guy is down, this guy will be down and not exactly the opposite. Just to make a long story short, and with the help of systems biologists, we find out that actually the reason because the expression of PER2 and the expression of P53 did not coincide is because there is a shuttling event that takes place in the middle. So PER2 and P53 associate in the cytosol, they move to the nucleus, and then they dissociate in the nucleus, and that dissociation in the nucleus results in a delay by the time that P53 is degraded. And we discovered all this with the help of systems biology. The philosophy in my lab is very simple. I always tell my students that what is important to remember is that you need to have a really interesting question. And whatever it takes us to answer that question is on the second plane, right? I mean, we just want to be sure that we have an interesting question. If we need to learn something from other disciplines, we would just bring them together to help us to answer that question. So that's why we use systems biology in this approach, and we use structural biologies and biophysics to answer some other questions. So anyway, so we later proved that that PER2 actually not only binds to p53 and prevents p53 degradation, but PER2 helps to move p53 to the nucleus, pumps p53 to the nucleus, and then it dissociates from this complex. And when it dissociates from this complex, it has a transcriptional activity. And if it's not needed for the transcriptional activity, it goes back to the cytosol where it's degraded. And this is what it causes the delay. And that's why the phases of the presence of PER2 and p53 are shifted. So when we thought that we knew pretty much everything about the interaction between PER2 and p53, I have a really cool conversation with a very clever graduate student in my lab, Jin, and she has been for a really long time trying to convince me to get into the business of ubiquitination, which I managed to skip for a little bit. And then she brought to my attention a number of publications that state that the way that the stability of PER2 is managed in the cell, let me go back. The way that the stability of PER2 is driven by phosphorylation in the cell cannot be the only mechanism. So she basically gave me one day a pile of papers like this and say, Carla, if you analyze all these papers together, this is telling us that there has to be another way to degrade PER2. Why is this important? Because if the degradation of PER2 happens in another way, then that means that there's more than one mechanism to control the length of the timing in the cell. So I told her, well, be my guest. I mean, I think you have a very interesting question, all yours. And that's what Eugene did. She was a very clever student, and in collaboration with another fantastic student in my lab, Xianlin Xu, they both work very hard to actually address the question of which one would be an alternative mechanism for degradation of PER2 that can control the clock. And for that, she uses a molecular approach. She found new interactors for MDM2. She used a biochemical approach. She proved that phosphorylation was not necessarily required for the degradation of PER2. And then she actually used a structural approach and a biophysical approach to determine which ones were the mechanisms and the point of regulations within the PER2 structure. Not only that, I think the cherry on top, I like to call it the strawberry on top. I don't like cherries, but the cherry on top was that she really find out that there is a way to control the length of the clock by modifying proteins involved in the cell cycle. So what she actually did is she modified the level of the negative regulator of PER2 by either overexpressing MDM2 or downregulated MDM2 and changed the period length of the oscillation. This was a fantastic discovery because sorry, I'm not very objective on this. I really think it was a really cool discovery because all the time we were talking about circadian rhythm controlling the cell cycle, and this discovery shows that the cell cycle can impact the length of the clock. And this has a lot of potential implications for therapeutics. So the model that we end up developing after all the work from Xiaolin and Jin is that is the one presented on this slide that, as you can imagine, is getting closer and closer to Jackson Pollock's canvas. And this is only with 3 molecules. Imagine if we add more. Okay, so now let's go and think about this for a second. The molecules that I mentioned to you are MDN, p53, and PER2. And what is particularly interesting about this 3 molecules together is that p53 and MDM2 are 2 molecules that they are highly mutated in cancer, okay? P53 is mutated in 80% of cancer. MDM2 has gene duplications in most of the cancers. There are cancers that they have 1,000 copies of MDM2. So as you can imagine, MDM2 and P53 have been targeted for therapeutics for a really long time. There are molecules out there, some of them in clinical trials and some other ones at the bench on preclinical trials that aim to either control the activity of P53 or control the association between P53 and MDM2. So because our circadian protein PER also interacts with P53 and MDM2, we actually start looking for, okay, is there any possibility that PER is also mutated in some of these cancers? And can this association between PER, P53, and MDM2 can be modulated somehow to favor the transcription of certain genes? So one thing that we found is actually, if you go to the Cancer Genome Atlas and look for all the genomes deposited there on breast cancer patients, and you just separate the ones that they carry mutations on the BRCA1 gene, because that will end up in the type of cancer that is inherited, versus the ones that shows mutations, the ones that they are breast cancer that comes from individuals where the cancer arises spontaneously, you will find out that among the 3,600 samples coming from the genome of tumors that arise spontaneously in these patients, a great deal of those tumors, they have mutations on the PER protein. Okay, and I'm showing you here mutations only on the coding region. There are other mutations on the regulatory region, but I want to bring to your attention that some of those mutations are particularly prevalent in different subtypes of breast cancer and in different areas, some of which affect the transcriptional activity itself of PER2 and some other one the binding activity. I don't know much yet the direct implications of this, and we can discuss it later, but what is interesting is that this particular region of PER2 is the one that is associated with controlling the clock, and this particular region of PER2 is the one that is associated with the tumor suppressor activity. So one way to analyze whether any of this will make sense from the therapeutic standpoint is to model and use molecular modeling and analyze how the interaction between P53 and PER2 happens. And let me tell you that the structure of P53 is partially known. There are some regions that they are not known because the flexibility of the molecule, and we couldn't get any particular diffraction that is reliable, so that part of the molecule is not known. And PER2 structure has been modeled here. There is not any crystal structure of PER2, and the reason is because PER2 is a largely unstructured protein. PER2 is literally an spaghetti. So what you actually see here is a model that we generate, and what you see is P53 wild type, and this is the helix that actually faces the DNA and control the transcriptional activity. And this is a loop that is critical for its association to PER2. What is particularly interesting about this interaction is that there is a mutation in this loop and a tyrosine residue that changes to cysteine that causes the repositioning of the loop and the change on the Krabis that is generated here. This repositioning makes the interaction between P53 and PER2 stronger, so stronger that very many of the drugs that are used now to reverse the structure of mutant P53 to a wild type form to be therapeutically active are not really working. What does it mean? It means that if you want to use that drug to reverse the phenotype of P53 mutant to wild type and had a detrimental effect on a tumor cell, meaning triggering apoptosis, for example, that drug will not have any effect if that tumor expresses PER2, or if you give the drug at a time in which PER2 is high in the tumor. So this brings us to what I want my fellow clinicians to start thinking about, and I've been trying to convince as many clinicians across my office for years, and it's to start are to begin paying attention to time of the day medicine. One day I was really mad about everything that I was reading and the lack of implementation, that I wrote this article that is called Chronotherapy, Intuitive, Sound Funded, but not broadly applied. And it's simply, the title was my mood that day when I wrote the article. I just want to bring to your attention the fact that there are fantastic research done by my colleagues published in top journals, and you just pick the journal that you like and you will find an article about circadian rhythmicity and the impact on the pharmacokinetics and pharmacodynamics of drugs, as well as the activity of oncogenes. And all of them, all of us that work on this particular area want to make sure that you that implement medicine have the resources to understand ways to implement this therapeutic or this approach in the clinic. Basically, what we want to do is to see if we can do better in terms of personalized medicine. We know that personalized medicine is the way to go. There is absolutely no doubt about that. The combination of genomics and the combination of all the omics that we have out there, plus tailored therapy is the way to go. What we want is to take into consideration that very many of those therapeutics can be delivered more efficiently at a specific time of the day. And I'm going to give you an example that comes from John Hogan's paper. This is a commentary on Science that comes from one of his original papers published in Proceedings in 2019. What John did is he picked all the clinical trials published in clinicaltrial.gov from 1965 to 2019 that has at least information about 2 times during the day in which the medication was given, okay? So there were only 105 clinical trials since 1965 where the drug was delivered at 2 different times of the day. And what you actually see here in blue is the result of his analysis that shows that in most of them, giving the therapeutic at a different time makes a difference. And that is particularly true for diseases like cancer or hypertension. And some of these are things that we, or you actually, apply in the clinic. This is particularly important if you consider the half-life of the drug. I mean, and we have studies now, this is just one very small study, but there are way much more studies that shows that the efficiency of time of day delivery goes even, is even positive for drugs that they have up to 15 hours of half-life. So the take-home message that I wanna pass to you is that maybe combining personalized medicine with what I call the 4th dimension of medicine, which is the time of the day treatment, can help to improve the efficacy of current treatment. And please don't take me wrong. I'm not saying that the time of day medicine is going to cure anybody, okay? But what I'm saying is that we can improve the efficacy of the treatments. We can have better protocols for delivery of combined drugs, okay? We can have, we can develop cost-effective prevention strategies. We can maybe even repurpose drugs. Oxaliplatin is a very good example of a drug that has been repurposed because results done in clinical trials using chronotherapeutics. And more importantly, I think we can improve the quality of life of patients. I'm going to close by giving you a couple of examples. If you go to the clinicaltrials.gov website, you will see that there are roughly 220,000 registered clinical trials in the world, okay? And most of them are actually happening in the United States. Now, if you do some further analysis, you will find out that among 220,000 clinical trials, only 348 actually include some form of circadian intervention, okay? Only 0.16% of all clinical trials currently ongoing in the world has any form of circadian regulation or intervention. Of the 0.16%, only 3% are related to cancer. Most of them, they have to do obviously with behavior and mental disorders and studies on the nervous system. So only 3% out of the 0.16% is any application in cancer. Now, if you consider that the pharmacokinetics, pharmacodynamics, tolerability, and efficacy of very many of the drugs, okay, that are used in the clinic for the treatment of cancer are controlled by circadian factors, then it becomes an issue. Now, if you are a person that do not work necessarily on cancer, but you are interested about this particular topic, I want to bring to your attention another fact, which is the fact that among the top 100 best-selling branded drugs in the market, okay, most of them target product of genes with rhythmic expression, okay? And you can pick the one that you like. This is the table also that comes from John Hagendash's work in the proceedings paper. And, you know, look, GERD, I mean, Nexium that is given for GERD is one of them, you know, bestseller drugs out there. It targets a circadian gene. I mean, these are all genes that they are targeted by these drugs that they have a circadian expression. If you know, the same way that I mentioned Nexium, I mentioned Ritalin, or just to be on the viruses, so Tamiflu, any of this, they follow, they target circadian oscillatory genes, even Viagra, okay? So for all of these molecules, for all of these drugs, there are a specific time of the day in which they should be delivered. Now that brings another problem, which is the problem of when to deliver things, but we can discuss that in a minute. So I just want to finish with this sad tone that only 4 of the 50 currently most prescribed drugs have FDA labeled time of day dosing recommendation, okay? Only 4 out of 50. And the 20th World Health Organization's model list of essential medicines makes no mention of dosing time. So we're basically wasting an opportunity to make treatments more efficient and reduce the burden that comes with side effects of drugs. So from the standpoint of my lab, all what I can contribute is with a molecular basis for understanding how those drugs work. But I really hope, I encourage you to actually take into consideration time of day medicine for further application. I'm gonna finish my talk here, thanking to the people that actually do all the work. My fantastic graduate students, Charlene and Jesse and Jinjin, and previously Lian, who has done a great deal of the work that I present to you today. Tetsuya is a former postdoc. He is now professor at Osaka University, and my collaborators, Jay Kim from the KAIS Institute, John Tyson, Anne Brown, and Diego Colombek from Argentina. Thank you, and I'll be happy to answer any questions that you might have. You're fantastic and giving me that applause.